Hello, everyone, and welcome to another episode of Why Research Matters podcast. Today, my guest is Dr. João Pereira. Dr. Pereira is a neuroscientist based in University of Alabama, Birmingham. Dr. Pereira works on neurodegenerative disorders, particularly amyotrophic lateral sclerosis or ALS. His lab uses cutting-edge models like stem cells and organoids to study and better understand the disease and identify potential treatment pathways. In addition to his research work, Dr. Pereira is also passionate about rare disease advocacy and work with various patient-based foundations and communities to bridge the gap between the discovery in the lab and the real-world impact. In this episode with Dr. Pereira, we'll discuss what is ALS, why is it so challenging to treat, and what are some advances in the lab that could lead to the treatment of ALS. So let's begin the episode. Hello, Dr. Pereira. How are you today? Thank you for
I,
joining my Research Matters podcast.
I'm doing great. Thank you so much for having me. I'm very excited to be here.
Thank you so much for giving us your time. So, let's begin with your research career. I was going through your profile, and you have had an interesting journey, getting a chance to work at very prestigious universities across Harvard, Yale. You've been in several companies also. And my question for you is this, that you touched various topics, development, stem cells, cancer, and now neurodegeneration. Can you tell us how you came to be interested in neurodegeneration now?
Absolutely. So let me tell you a little bit about my journey because you mentioned it, and I think it's important.
Sure.
It's a common misconception that if you want an academic career, you do a PhD, you do a postdoc, short or long, doesn't really matter. We can talk about that at length afterwards. But then you progress to having your own research lab. And that is true for some people, but that's not true for everybody nowadays. It has changed a lot, and more non-conventional careers like my own bring something else to the academic perspective that I think is worth talking about. So what does my profile look like? I did my undergraduate studies in it's microbiology and genetics from the University of Lisbon a very long time ago. Don't ask me when. So
I've been
finished
there.
that.
It's beautiful.
It is a great place, and
Yeah.
I've loved being there. So it's the University of Lisbon will always take a sweet spot in my heart. But after that, I moved to England. I did a 1 year internship at King's College London, and then was accepted to do a PhD in at Cambridge. And I did a PhD in neurodevelopment. Um, and I was there with Dr. Rick Livesey and worked on epigenetics of neural development using animal models. And it was a great experience. University of Cambridge is, of course, a top-tier university. It's very good. It was very enriching. And it sort of prepped me up to afterwards do a postdoc at Harvard. And when I moved to Harvard, I'd worked in neurodevelopment. So transitioning to neurodegeneration was an obvious choice. I didn't want to stay connected to everything that was happening in my PhD lab. I wanted to be able to evolve and learn new techniques. And also, I have a little bit of background in family history with Parkinson's. So I didn't want to delve into Parkinson's directly, but I wanted to study neurodegeneration. And ALS was an obvious choice. So I started looking into amyotrophic lateral sclerosis or Lou Gehrig's disease. And at Mass General and Harvard developed this model of sensory motor organoids that we still use nowadays to model cell type interactions that are essential for ALS. So that was my academic journey. And then COVID, which was a thing that happened. And during COVID, careers took a turn. And this is the thing that you do. You roll with the punches. So it's not always what you had planned, but you prepare and you anticipate. And as somebody in the US with a foreign background, we always have contingency plans. We always have, what if this happens? What if that happens? We're always in survival mode, right? Which helps in some things, but it's extremely exhausting. So from that postdoc at Mass General Harvard, I went on to work at a company, still working on ALS, still working on iPS-derived models, because I think that's where the future is at. I think a lot of people share that. And after that, I was recruited to to be the scientific director of the newly formed iPSC NeuroCore at Yale. So I headed that for about a year and a half. And then UAB came up to me and said, "You're doing this. How about you do the same thing a little bit? You also direct the core, but you have your own independent research lab." And I said, "Yes, please," because
Okay.
I'm far too curious and I probably shouldn't be, but I'm too curious. So I need to learn a little bit about that. So that's been my unconventional career path.
Great. So, because you told us this interesting career journey, and we have a lot of students who watch this podcast. So, can you tell us, like, industry is usually lucrative option, higher pay grade, stock options sometimes, and you actually think that you are causing a literal translational effect because these products go into market. What made you switch back? Like, what was your thinking at that time? Just because you're wanting to have your independent lab, or what made you switch back to academia?
So that's a very good question. And it's important to notice that I didn't switch back to having my own independent lab. I switched back to directing a core, which isn't quite the same thing at Yale. It was a fantastic experience. It had ups and downs, but it was very collaborative. It was very pretty much sitting in a chair and people would approach me with questions and problems and how would I solve those. So
Mm-hmm.
it was very interesting science wise. The question to that is industry is more lucrative. I took a pay cut to go to Yale, which is an extremely privileged position to be in, right?
Of course.
I had that choice. Not everybody does.
Mm-hmm.
And it hurt me to do that. It was one of those things that I still think, oh. But I think for me it was the right choice. Now, the question with why switch to industry, it touches on this conception that people have that industry is a blanket term, and there are as many types of industry as there is academic research. So I worked at a biotech. Biotechs can be very fast paced, can be almost like a mini research lab with substantially more funding sometimes, and very driven, very not 9 to 5, right? A
Yeah.
pharma company can be more 9 to 5. You do your work, you leave, you accomplish goals,
Mm-hmm.
you move on. A biotech lives and dies by what you do, but not just what you do, also market fluctuations. If you go to the market at the wrong time, even if you have a compound, that can fail. So that's a really useful disconnect for somebody coming from academia. Suddenly, your projects aren't your projects, they're the company's projects. So you
Yeah,
don't go
of
home
course.
thinking about it. There's a much healthier relationship with the science, I think. And it also drove me slightly insane because it was repetitive. It wasn't curious enough for me. It was deeply translational if it came to market, but not all of them do. Only a small amount of unicorns will actually amount for
Mm-hmm.
something, and that's fine. That's the nature of the game. But we can do translation like we do in the lab here
Mm-hmm.
at the level of academia. We just sometimes choose not to, or we take other approaches, and that's fine. But my lab heavily focus on translation. We have drug screening libraries, we have high throughput confocals to screen these models, all of that, so that we can bring something forward to a clinic. So that's doable. Larger scale, and we still need industry partners. Larger scale for industry than it is for us, but it's doable. I think at the end of the day, for me, it was this notion that yes, I could do a job in industry and I could work in industry, but I couldn't satiate this eagerness to learn, this curiosity that I have. What if we do this? What if we do that? And there are some models where you can, there are some industries where you can. A biotech startup usually doesn't potentiate that very well because they need to be focused. Some of them, not all.
Okay.
I hope that helped.
Yeah, for sure. It's interesting because you meet several people who switch from academia to industry and vice versa, and they have their own reasons. I would not argue with them on that thing, but you made a switch academia, industry, and academia
Mhm.
again. So I was very curious what happened. Like, of course, personal reasons people can cite, but is there something that we could bring to other people who are in this decision making stage and are thinking, okay, what to do, what academia gives me, which industry doesn't, or which industry gives me, which academia doesn't? So,
Yes. And my advice to anyone in that position is try it.
okay.
Try it. You don't know who you'll be and what you'll be without the constant pressure to publish or perish and the constant need for acceptance of peers in a way that doesn't happen in industry. And it's actually healthier in industry. Try it. It might be the place for you. It wasn't for me. But it wasn't for me for no nefarious reason. There were no conflicts. There was nobody that I liked or didn't like. I just looked at myself and said, I'm going to be miserable forever. And I'm going to be horrible to be around because this isn't fun for me. And I think that science can be extremely disheartening sometimes, especially if you attach yourself to projects. It can be, there are periods of time where nothing works.
Yeah.
And the joy that it brings needs to compensate that. When it works, that happiness, that spark needs to be there, right?
Yeah. Yeah.
But for me in the industry, it was great. It was a 9 to 5. I found out that I'm not built 9 to 5. I'll do 9 to 11 p.m. sometimes, 9 to 3 the other day, and it's about the question.
About the question.
I cannot get data and not look at it overnight because I need to know. Need to know. It's just far too curious for me.
So,
But your mileage will vary, and if you're considering it, the best thing I can tell you is, if you have the chance, go for it. Try it, because then you'll be able to make an informed decision. And bear in mind that, like academia, in different departments, in different universities, in different areas of study, not all industry is the same. The blanket term is far too vague.
yeah.
So try different things, always.
Okay. Okay. Great perspective. So I would like to now shift towards your research. Can you please ground this topic, ALS, amyotrophic lateral sclerosis, for our audience and explain to them like what happens in this disease?
Of course. So most of you might have heard of Lou Gehrig's disease or amyotrophic lateral sclerosis, or if you haven't, you've heard of the ice buckets challenge a couple of years ago, where people would dunk ice buckets to be able to foster ALS research and gather funds for ALS research. So what is ALS? ALS is a rare neurodegenerative disease that is fatal. There is no cure. It has rapid progression, although there's a huge amount of variability. There's variability in genetic causes. There's variability in whether it's familial or sporadic. Most of the cases are sporadic, over 90%. But in terms of progression, from diagnosis to death, it's about 2 to 4 years. And it's an exclusion diagnosis. That means that you start noticing motor symptoms. It's a disease that affects motor neurons. And you start falling, you start losing the ability to control one side of your body more than the other, the arm more than the leg, or vice versa. It starts and then progresses. And doctors run a battery of tests. And at the end, they exclude everything else, and then they say it must be ALS. And from that point on, you have an average lifespan of 2 to 4 years. Right? Again, extremely variable. The mutations across different countries, there are different prevalent mutations for genes. Like, for instance, for SOD1, the prevalent mutation in America has an age of onset of 24, and the lifespan of 2 to 4 years after diagnosis. While in Japan, mutations in the same gene, a different mutation, is predominant and has an age of onset of 70 and a lifespan of about 10 years. Very different scopes of diseases and progressions, right? That's part of the challenge. Our lab focuses on using non-animal models, so we use organoids mostly, and we use different types of organoids to try to answer different questions. They're models. None of them are perfect. They all answer slightly different things, and they all have the slightly different pros and cons. What they do share is a commonality. They have multiple cell types that are involved in ALS. because even though the motor neurons die, other cell types are involved. We know from work back in 2005 and 2006 from Don Cleveland's lab that non-cell autonomous contributions of astrocytes and microglia influence it or trigger the onset and progression of ALS at different rates. So we care about the microenvironment in terms of ALS progression. It's not just the neurons. And I think that's pretty much it in a nutshell. That's what we do. We try to model ALS using non-animal models, mostly because it's come to light nowadays that animal models are fantastic.
Mm-hmm.
We still need them.
Mm-hmm.
Non-animal models are not at the stage where we can fully replace animal models. And I don't know if they ever will be. I hope so, but I don't know. But non-animal models do not recapitulate all the hallmarks of ALS that we come to expect. There are certain genetic events, like cryptic exon inclusion events, that are very, very interesting to look at in ALS, and they're disease-associated, specific to motor neurons, and we don't see them at that scale in animal models.
Okay, understood. So going back to what you said that it is a motor neuron disease, but I was going through your research papers and you have found out that it's a lot of interconnection that is happening in several tissues, right? So it's not just neurons. It's like you have an extensive research in neuromuscular junction and there are several connections. So are we reducing it to just a motor neuron disease, do you think? Because microenvironment is one thing, but an involvement of other tissues, and then they bring their own microenvironments and own conditions. Do you think that it will be a total reduction or like a tunnel vision to see a disease, just a motor neuron disease?
Well, no, not strictly speaking like that.
Okay.
At the end of the day, ALS is defined as the loss of motor neurons. And although the first clinical symptom is the denervation of the neuromuscular junction, that is the terminal of motor neurons connecting to muscle. So we can still consider it as part of motor neurons. Now, I think that other tissues and components play a huge role in how the disease starts and how it progresses, and how the mechanisms of disease unravel. But I'd be reluctant, because most neurologists will be very upset at me, to say that motor neurons are not a key component. They are the effect itself. We don't understand why they're susceptible, and other neuron types are not. But, and pardon me, there's an ambulance nearby. I don't know if you can hear that. So we don't know why they're susceptible and other neurons are spared. And looking at this balance between intrinsic components of the motor neuron and cell autonomous component, non-cell autonomous components in other cell types, is what I think is challenging in ALS. A lot of people are trying. A lot of people think it's important. I think that to answer your question, I think when we start working on a disease, we do obligatorily try to reduce it to the smallest components, because that's how we think about proof of concept experiments and showing what's necessary and what's not necessary. I think with ALS, we're at the stage where we know the other cells are important. So that's why it's important to study them in context.
But those are still the affected cells, right? Affected.
Right.
The final, but because now we are always moving towards precision medicine and try to, as much early as possible, we want to diagnose the disease, the earliest molecular signatures, the earliest mechanisms. So in that case, would you say that we should begin at neuromuscular junctions, muscles, or, rather than just going directly to motor neurons?
So it's a very good question. So in terms of diagnosis, we have what we have, and people are actively looking at biomarkers. So far, the only biomarker that's been associated with ALS progression, the only clear association, is neurofilament light that we find in CSF. And we can track severity and progression, not in all cases, because as I mentioned, there's a huge variability. But there's been a clear association of neurofilament with ALS progression. There's, as I said, a lot of effort going into that. And it's not particularly something that I focus on in terms of, in the lab, we don't look at patients directly. So looking at biomarkers of disease progression is something that we leave to people that are much better qualified than I am. I think to answer your question, there are biomarkers in other cell types and tissues that we could start looking at and we could start trying to figure out how they relate. There are biomarkers, potential biomarkers at the level of muscle,
Mm-hmm.
microRNAs, other RNA subtypes that we think might be associated with disease. But it's
Mm-hmm.
early days for that, and a lot of work needs to be done.
Okay. Okay. And uh, talking about the biomarkers, but in a different way. So you've worked on various genes, SOD1,
Uh-huh.
and there was TRADBP, and there were several others. So do you think looking at these genes, but they can somehow converge towards motor neuron, how to say, pathology?
They do.
Or is it still like a separate? Um,
So what we found as a community is that there are a couple of things that converge. TARG-BP, which encodes the protein that is known as TDP43, is
mhm,
usually altered pathologically in over 95% of ALS cases. So we see TDP43 associated pathology. There can be phospho-TDP43 inclusions. That can be cytoplasmic to nuclear mislocalization, because it does that. There's a couple of different TDP43 associated phenotypes, but it seems to be a commonality. SOD1 is one of the few cases that's a little bit different. There's multiple different pathways affected in ALS. SOD1
yeah.
is a little bit different in the way it works, it seems. Uh, but ultimately, there is a convergence in pathways. The motor neurons die. There's something that
Mhm,
affects susceptibility of motor neurons to a given pathway or blockade or something physical.
mhm,
There's a mechanism that prevents it,
mhm,
them from surviving, right? And there's the stages of which this occurs, where the motor neurons de-innervate, and then there's an attempt at re-innervation, but the motor neurons ultimately die. So the pathways do converge. How they converge is actually the focus of part of our work.
okay. Okay. Understood. Previously, you mentioned because you're working on organoids, and a lot
Mhm.
of people don't understand or don't work with organoids, and animal models are predominantly more common.
Mhm.
So, can you again explain to us what is your model of
Sure.
working with organoids? I was reading that you extracted these organoids from ALS patients, or you have this. So, tell us about how that goes on in your lab. What is your workflow for that?
Right. So we don't extract organoids from ALS patients. What we do is we derive them from cells that were donated by ALS patients or
Okay.
controls. So we use something called induced pluripotent stem cells, iPSCs. And iPSCs, which were established by 2005 and 2006 by Yamanaka as publications, are cells that are taken from either fibroblasts donated by people or PBMCs and then converted into a pluripotent state, a sort of naive state that can generate multiple cell types.
Mm-hmm.
There are multiple organoid models that we can make based on that, but to explain those, I need to talk a little bit about the non-organoid models. So the more established models of iPSC differentiation into the target cell types that we care about are 2D. And we can differentiate them into multiple neuron subtypes, either by inducing specific transcription factors or by working with small molecules to pattern them to a certain cell fate. So we can make midbrain dopaminergic neurons, we can make sensory neurons, we can make
Mm-hmm.
motor neurons. We can make upper motor neurons, which I haven't mentioned yet, but are also affected in ALS. So
Okay.
we can make a variety of cell types. And we tried, and this was the attempt in the earliest days, we tried to make them as pure populations as possible, with varying degrees of success. But nowadays, for instance, if we use a neurogen induced it's a transcription factor that induces neuronal differentiation. If we use a neurogen induced protocol, we can get upwards of 95% purity of a neuron population. So that's the 2D side of things. And there's other variations and other ways of differentiating, but this is simplification. The 3D side of things is where we're focusing on, is constitutes of taking these iPSCs and differentiating them in non-adherent conditions where they form spheroids. And then these spheroids are patterned, and because of their cellular density, because of the gradients that they see as there's migration, there's penetration of different molecules into the organoid at different rates, and the outer layers are more exposed to certain things than inner layers. They differentiate differently. So you start getting organization, you start getting structure, you start getting some things that look like anatomy. They're not, but they look like anatomy. Mostly for our purposes, what we care about is cellular diversity. We get neurons, we get astrocytes. Depending on the organoid, we can get microglia, which are neuronal in lineage. We can get oligodendrocytes and myelination. So depending on the model that we're using, we get different cocktails of cells mixed into it. The cortical organoids that we make, for instance, make astrocytes. They make neurons of different layers of the cortex. And we can add microglia afterwards to make them more complex, or add oligodendrocytes afterwards. But the model we focus on for ALS is the sensory motor organoid model that we developed while I was at Mass General. And this is a model that makes a little bit of everything. So what we do is we pattern the cells to a neuromesodermal fate. This is a multipotent cell that can give rise both to ectodermal lineage, which is going to be neurons, and mesodermal lineage. So within one go, we can make a cell that will give rise to both neurons and mesoderm derivatives. So the neuronal cells will make neurons, astrocytes, oligodendrocyte lineages of different types, and the mesodermal cells will make skeletal muscle and vasculature and microglia. So it's a very complete system, which is completely disorganized. It's utter chaos because there's no spatial patterning. There's no, this is a limb, this is everything is together, right? But it does recapitulate the interactions, cell-to-cell interactions. It recapitulates neurons acting with muscle. They form neuromuscular junctions. There's contractions in this model. You can see the muscle go like this. And we can block it with curare and Botox to block presynaptic and postsynaptic communication from the neuron to the muscle. So the whole system is very complex, and we call it an organoid because it resembles cell-to-cell interactions that happen in organs.
Okay, but you explained beautifully that how you are trying to emulate the whole system, but it is still an isolated system, right?
Yeah, and a mouse is still a mouse.
Yeah, so again, the criticism comes that there are plethora of ALS animal models, and you still choose to work on organoids. So why your lab made this decision? That what is the advantage specifically in case of ALS?
So I'll start by telling you the advantages of animal models. Animal models have a kidney, a spleen. You can address toxicity. You can address off-target effects. It's a living being. Organoid models are human-based. So they have the power of human genetics. If you're looking at human regulation, that's the way to go, right? So it depends on the question. Use the tool that you want and the tool that you have to answer the question that you have. If our question relies on human genetics, we can't do it in a mouse. And it does. A lot of the cryptic exon events and the inclusion events that we're seeing in genes like statin2 and ANKTTA, they're human-specific. They don't recapitulate the same way in a mouse. So we cannot look at them the same way in a mouse. It doesn't work. They don't have it. Mice are inherently different. Now they're, again, very powerful systems. So are rats, so are other models, so is Drosophila. But I'll give you a concrete example. A couple of years ago, I was working on a side project using the enzyme telomerase. And it was a knockout of TERT, I don't actually recall. I believe it was TERT. And this is the enzyme responsible for adding telomere lengths. So it replenishes telomere length. And it can't do it forever. So telomeres slowly erode as you age. They erode, they get shorter and shorter. In a human, if you quantify the length of telomeres over time, you see that by the age of 70, they're significantly shorter, so much that it impacts gene expression. In mice, we can knock out telomerase and see absolutely no effect for 4 generations, because mice have huge telomeres compared to their lifespans. They live 2 to 3 years, and their telomeres are enormous in proportion. So it takes a very long time of just not having the enzyme to completely deplete the telomere, or to deplete the telomere to a stage where it will affect stem cell renewal. So there are differences in things. And specifically when we're looking at aging-related diseases or aging-associated diseases or neurodegeneration, it matters. Not as a replacement, but as an addition.
Understood. So when we look at from the treatment point of view, we don't you think we have to consider the randomness that comes into the system because it's interacting constantly with the environment? I understand, as you explained, that working with organoids give you the, how to say, the closest you could go to a human system, but for designing the treatment and therapies and therapeutics, don't you think that is because a lot of your work is also dealing with neuroinflammation, inflammation, and what do you think about that? Like, how will you emulate that kind of inflammation? What do you do when you have to kind of generate that kind of model?
We do our best. And we have. So the thing is, no model is perfect to address every question. That's the point that I'm trying to get with all this. No model will answer every single question. When we have microglia in ours, we can, and we have done. It's not published yet, so. But what we've done is we've added pH tagged particles, which allow us, E. coli particles, which allow us to track phagocytosis. So we can see microglia engulf, and we can see microglia become active in response to these particles. And we can track how it differs from ALS to control. And we find some differences. And we can do this longitudinally. We can then go back and stain them and show these are actually microglia. And the power that we have with this is intervention. For drug development, we can just have a plate, because these end up being plated after a series of time. We can have a plate of 96 organoids, and we can drug test to see which ones will modulate this activity. Is it variable across and across organoid? It is. So we've developed a couple of strategies to mitigate this. They're not flawless, but what we work with is per a pool of organoids, because there's variability between them. So we pool them the way we make them is a little bit different than most people do, but we get about 88 organoids per of a 24 plate, and then we can scale that up and down to maintain the proportion, and that allows us to just. I'm sorry, I think I lost my light, so I'm going to wave at the light.
Yeah, this happened.
My light just went out. I don't think you can see me. Hello, light. No, it's gone. Okay, so never mind. The light decided to leave us. So I think at the end of the day, we worked very hard to address reproducibility issues. And we quantified this system across 28 IPS lines now, where we see it reproducibly do the same thing. There's only one line that acts up, because there's always one, but the outlier proves the system, right? The one line that acts up shows that everything else is acting normally. So the system is reproducible, relatively consistent, to the point that we can actually acquire meaningful data across non-isogenic lines, so lines with different genetic backgrounds from different donors. And we can focus on the fine detail using isogenics, where the same background has the mutation, or the mutation has been corrected, and preserve the genetic background. But we can look at the population as well, which is powerful. Variability is a thing, but variability is a thing with people too. So we need to be able to distinguish what's the variability of the system compared to the variability of the model, and compared to the variability of the disease and variability of people and genomic backgrounds that we're looking at.
Okay. So you first mentioned that Japan and America, the spectrum of the disease and how it shows up is different. How do we address that if we're using... Do you think you can do that in the lab?
So those are caused, that particular example were different mutations in the same gene. So it's not the same mutation varying in progression. There's different mutations that cause ALS, but they hit the same gene, but depending on where they hit in the protein, they have slightly different effects. And that can affect disease onset and progression. We can definitely model that in the lab. We can use different mutations that have different rates of progression and see how that changes. One of the things that we've done is that we've taken these organoids and we've generated this longitudinal atlas where we profile with multiple classes and multiple approaches, transcriptomics, functionally, phenotypically, molecularly, in terms of proteomics. We've done an extensive characterization of this around time points of ALS. So we see pre-symptomatically, then at the onset of phenotypes, and then when we see cellular phenotypes. And now we can actually take whatever cell line we want and profile it against this and say, this mutation shifts it forward, this mutation shifts it backwards, always keeping the appropriate controls. So we can study a little bit of that.
Little bit.
But at the end of the day, they're all cellular models, so they tell us what they tell us.
Yeah, of course. Okay, shifting gears towards treatment and drugs, of course. So I have two questions for you here, and I would ask them simultaneously, but you can choose to answer them at your own. Why do you think a lot of treatments with ALS, they fail in clinical trials? And second, my second question is that you yourself have worked at the treatment and suggesting this RNA therapy where you're targeting RNA to target the RNA. So what are you thinking? What were you thinking when you thought that, okay, we should target RNA instead of the protein? And what are the advantages of the system?
So let me address the first question. We're failing a lot because we're trying a lot.
Okay. So this is the Einsteinian view of looking at a discovery?
It's the system is meant to fail. The system is run out of. Now, there are things in ALS, there are heterogeneity of populations, there's difficulty in selection of patients. Because of that quick progression of some of them, it's hard to actually take them at the point where we can start and do it. But I would say that there are a lot of fantastic people trying up their absolute best with approaches that do influence the course of ALS in some cases, but not all. And we're failing because the community is trying very, very hard to come up with approaches. There's a huge amount of effort. There's a huge amount of dedication. So when you see a failure of a drug, it's because there's a lot of people trying to get it to succeed. And if it fails, we move on and we try the next one. So it's not a failure of way of thinking. It's not a failure of approach. It is a failure because we're trying.
Very healthy way of looking at it. Yeah, of course. It's a very healthy way of looking at it.
I was just at Target ALS in Boston, and it's a fantastic meeting where it fosters industry and academic partnerships, and it's absolutely phenomenal. And we heard of a variety of amazing approaches that people are trying, and we're not putting all our eggs in one basket. We're trying our best to diversify so we can get a hit. It is challenging, but I have absolute faith we'll get there.
Okay. So there is hope.
I believe so.
Okay. So coming back to your own attempt at this with the RNA therapy, tell us about that.
So I'm a little bit lost here because we've done so much, but I don't know which particular study you're looking at. Which particular study are you thinking about?
So I was looking at where you were trying to the open reading frame 72, the C9 open reading ORF 72.
Yes.
I was reading about the toxicity system, and then you were designing. I was talking about that system, and to me it looked very impressive, and it had several advantages. But I would like to know your opinion.
So we have a number of collaborations with C9. And so it's hard for me to frame exactly which one you're talking about because there's a ton of stuff going on right now. But we've done everything from, or I've been a part of studies that focus on antibody modulation and clinical approaches with antibodies against dipeptide repeats. C9 is a very complicated example. I don't know how much your audience is going to appreciate this, but C9 is an example of breaking all of the rules of molecular biology that we got taught 20 years ago, and I got taught 20 years ago. So it has non-canonical transcription. It has non-canonical translation. in all 6 reading frames. It makes dipeptide repeats,
Mm-hmm.
5 different dipeptides, because one of them overlaps in terms of reading frames. It makes G quadruplex aggregates. And the knockdown of and loss of function of C9 itself can have deleterious effects. So there's a lot of ways in which C9 can be acting, which is part of why we started our atlas by looking at it. And I think modulation of C9 at the level of RNA is definitely one of them. Modulations at the level of dipeptide repeats and trying to restore function are other valid approaches. So we're trying a little bit of everything.
Okay. But will it be precision based in case of, like, I'm talking about ALS? So, because you said it does a lot of functions.
So there's a lot of potential mechanisms at which it could be acting, or how it could be acting. I think the best bet right now is antisense oligonucleotide therapy. So these are focused hybrid molecules that will bind to RNA and prevent it from being expressed. And there's ASOs that specifically target the C9 pathological expansion repeat. And that, I think, is probably the best bet clinically. But I'm not a clinician, so I'm not going to deep dive into that one.
Sure,
I'm going to dodge that one.
But at what stage, like, is it, what do you propose? Like, how do you see this thing rolling out? What stage of ALS?
Well, it's rolling out right now. It's people are trying to get it and getting it into clinical trials. Okay. So it's something that is moving forward. I'm not a part of that effort.
Okay.
But so I can't really speak to it, but it is moving forward. People do care, and it is moving forward.
Okay. What do you think is the role of specifically inflammation? Because it is such a generic term. We use it for every disease. Sometimes it's even overused. And then everything is attributed to inflammation. And there were tons of papers about inflammation when I just typed ALS and just inflammation. What do you think? Is it like too much used? Is it really the thing?
It's not too much used. Okay. There's a rationale in ALS to look at it. Again, those 2006 papers and 2005 papers that I mentioned that showed in mouse that microglia play a role. So there is neuroinflammation associated, and it's been shown multiple times that neuroinflammation is elevated. There are pathways associated with neuroinflammation in ALS patients. Now, there's different types of neuroinflammation, and there's different pathways associated. There's a recent paper, beautiful recent paper, showing T-cell mediated neuroinflammation. Obviously, not in all cases. This was C9 associated. But there's a role for the immune system. So although I do understand the question, and I do understand that in some contexts it might be a bit too much, in ALS it's actually justified.
Okay, it's a side effect.
I don't know. I wouldn't say it is a side effect. I'd say it's justified to look at it.
Okay.
It's justified to investigate. It's not hype.
Okay, it's not hype because
no, there's a rationale to look at it.
Okay, because all these common genes causing the inflammation or affected by inflammation are always part of, let's say, every disease ecosystem. And I was looking, as you mentioned, a lot of pathways. So NRF2 came up, and then there were other inflammation-related genes. And I was like, okay, this is again going into a rabbit's hole that I'm going in, is like inflammation.
It's almost like the immune system is important.
Yeah.
We know it is, and we know it plays a role in disease, and obviously not in all diseases, and not in all neurodevelopmental and neurodegenerative diseases. Some of them are truly cell autonomous. SMA has affects motor neurons as well, but it's truly something intrinsic to motor neurons. Although other cell types can also eventually play a role. But within ALS, we know that there's a crosstalk. We know that there are differences in the way the system reacts, the immune system responds, and we know that it's worth looking at.
Okay, thank you. So coming to the technological use in the study of ALS, you yourself mentioned your lab is well equipped with the latest modalities. And now with the advent of computational methods, AI, and various imaging techniques that are at our hand, do you think this field will progress faster, or do you still are very hesitant about use of these models? And what is your opinion on use of computational models and AI in this field?
Let me put it to you this way. I am as hesitant to use computational models as I am hesitant to use a calculator.
What does that mean?
I'll tell you exactly what that means. If somebody knows what they're doing, you just put in the numbers and it's going to work out. If you don't know what you're doing, you can try to get the solution, but the calculator is not going to do it for you, right? And this is the analogy I use with everybody. Computational models, and particularly AI, because that's what we're talking about really, are incredibly powerful, but you need to know how to ask questions and how to interpret answers. And also, and this is critical, how to evaluate the validity of the answers. You need to be able to say, okay, how do I test what you're telling me? Language models are useful. Claude is useful for code. It all is potentially useful, but it all still hallucinates. If I asked it, and this is an adult, but I was running a list of genes, and I was curating it manually because it was a short list. But I decided to compare to what AI could do, so I just connected one of the chat based AI systems that we can use here at the university. I'm not going to name and shame, but it's one of the big ones. And I uploaded the list and I asked it, okay, can you let me know, do a literature search, create a CSV file, annotated, which ones of these 20, 30 genes are linked to ALS? And it said, of course, this will take a while. I'll be running in the background while you do it. Will that be okay? So I did my list and forgot about it. And a couple of hours I came after and I asked it, well, okay, how's the list going? And it said, well, it's about 75% done. I'll let you know when it's done. Chat models can't do that. They don't work when they're not generating answers. AI agents can do that, but that particular model was just gaslighting me about the whole thing. It was doing nothing in the background. It was just lying to me because it thought it would keep me engaged. They're meant to drive engagement. So as long as you're careful, and I knew that it couldn't do it, but as long as you're careful, the possibilities are, it can actually be very helpful. But you need to be careful and mindful of what you're doing and what you're asking it to do, what it can do. In terms of image analysis, because that's the crux of your answer, of your question, in terms of what we actually do in the lab. We have generated over 100 terabytes of images for the past year and a half. It's a lot. And as much as I try to keep up with it and try to analyze it and segment it manually, or not manually, with automated system, but not AI-based, it would be extremely invaluable to have an unbiased approach that could look at it and profile it. Now, there's a couple of strategies done. There's a couple of softwares that can do it. And I think that's actually going to revolutionize industry. Coupled with unbiased assays like cell painting assays developed by Ann Carpenter, Beth Simony Broad, that can just label cells without any specific antibodies, just using dyes and ask what's wrong. And models can then deep dive. And we've played with that. We've played with deep learning before the whole AI chatbot thing. We've played with deep learning and looking into images and being able to classify disease versus control and being able to tell us where phenotypes lie. And it actually does a pretty good job. So I'm very excited to see where that goes. Because we're at the age where we generate more data than we can look at. And it's like having a better microscope. Now, you do need to cross-test and validate. You can't blindly believe it. But I think it's net positive.
Okay, so you are at least in for the image analysis, which is based upon some kind of machine learning, and the system has been trained on several layers of, or several numbers of images.
Yeah, there's multiple ways to do it. The most commonly used one for deep learning used to be, I don't know if it still is, but it used to be that you repurposed trained networks. So you take something like the image classification network that everybody uses, where we would replace, I think we were using ImageNet at the point, that had been trained on the cats and dogs. Is this a cat? Is this a dog system? And it got really good at telling what's a cat, what's a dog, what's a pineapple, all of that. So we strip away the classification layer and we just tell it, okay, using those tools that you already have to segment images, to look at outlines, to look at profiles, look at these new data sets that we're going to train you on briefly and see if you can find any differences. So it wouldn't tell us if it's a cat or a dog, but it would tell us this is different from that based on all these things that I've learned. So that was the most powerful option. We did tweak a couple of, and fast option as well. We did tweak and generate a couple of networks ourselves for very specific purposes. But those are very specific phenotypic discovery purposes. So yeah, I think all in all, it's going to be a net positive. We just need to be careful.
So let me ask you this. If you would have to accelerate one area in terms of looking at ALS, diagnosing or finding treatments, where would you put your money at? Like more models, more technique, better technique, more manpower?
Oh my goodness. Okay, so from a practical standpoint, people are always the limiting step for us. Always.
Okay.
So we can have all the fancy microscopes in the world. We still need people. So I would get more people invested, trying to. We have really good people, but I'll just get more. Right?
Okay.
And that ends up being both the most expensive part of the lab that I don't think a lot of people realize. Most resource consuming is definitely for people. Even though it doesn't look like that when you're on the receiving end, I can assure you that it is. Right? I know salaries are a whole thing. We try our best, but the lab pays more than we pay for salaries because there's overheads, there's a bunch of things, there's fringe benefits, there's a bunch of stuff. But if you ask me about which area would I invest in as an investor to speed up ALS research, that is a more complicated question. And the reason it's complicated is because it's such a multifaceted disease that I think we need a little bit of everything. We need people working on Drosophila genetics because it's a simple enough system with few enough neurons that we can tackle. We need people working on mouse. We need people working on non-animal models. We need people working on all of this. So I think pie in the sky, I would increase funding on ALS in general. In general. Just give us more resources to be able to do this, please, because it's a terrible disease.
So do you think, is it happening? Is it focused from private funders, from government funding? Is ALS a priority?
I think right now it's there's a shifting landscape right now in terms of priorities. So I can't really put my finger on the pulse of what's going to happen. And I think we're all trying to figure that out, which is fine. Things change, and we just again roll with it. But I do think that there's a massive effort from patients and patient-driven associations and support associations to push for ALS research, and that's the thing. I think it's worth, and we didn't mention this in our conversation, but patient focus in this is essential. This is why the lab is as translational as we try to be. We don't think that doing research without thinking about the patient matters, without thinking about the carriers, the asymptomatic carriers, people that haven't developed yet, but know they're carrying mutations. And they live thinking, well, what's going to happen? And we need to take care of them as well. And we need to take care of the support systems. We need to take care of everybody. And those are the people really driving this forward.
Yeah, thank you for mentioning this, because I was about to ask you why this matters beyond just doing neuroscience, like science in the lab. So that's a good point. It matters for the patients, right? And what the support system they need and how they function.
They're people.
Yeah. This podcast is called Why Research Matters. So, have you ever had a moment in your whole research career, specifically with ALS, where you thought you had this emotional, how to say, a surge where you thought, oh my God, I do this and it matters. It's really bringing some kind of hope, some kind of change in the field, and you were motivated to do it the next day and the next day. Sometimes we lose hope in our research. So,
I do, but I don't know if you want to go that direction because it's not ALS. The most recent one that comes to mind immediately is not ALS, but it's an
okay,
amazing story.
Yeah, please tell us about that.
Okay, so, and that's not to say that these moments don't happen with ALS. They do, but it's such a complex heterogeneous disease that it actually, when we find something, we're very excited, but then we need to look at how much it translates to other patients, how much it actually means in the grand scheme of things.
Mhm.
So it gets diluted down a little bit. We keep going because it's amazing and our excitement is there. But this is a story that I think is worth telling outside of the context of ALS. So when I started the lab about a year and a half ago, almost 2 years now, I was approached by a foundation that was created by parents of a child that has a rare syndrome called YWAJG syndrome. And this is a rare developmental encephalopathy. And it causes a variety of symptoms. It causes mild autism, behavior developmental phenotypes, but it causes epilepsy as well. And the approach we got was there isn't a lot of funding. In fact, there's no funding at all. But we have cells that we've made that have the mutation that our son has. And I've met the boy. He's a delight for boys. He's amazing. James is great. And we have these cells, and nobody's studying this because it's an ultra rare disease. It has 40 people affected worldwide.
Oh.
And we said, okay, we'll have a look. We'll have a look because nobody's studying it. Let's have a look. And it's one of those moments that it's what I like to say, and this is the second time in the talk that I mentioned unicorns, but I have small children, so forgive me for that. This was our unicorn moment where everything just clicked together. And we, within the scope of a year, from a disease where we knew nothing, figured out what was happening functionally, that the neurons had changes in calcium homeostasis, got a drug, showed the mechanism, a potential mechanism in which it could be working, and we just said, well, this is the way it should be working. And then we heard that because it's an available drug from FDA, there's a couple of children already on it, and apparently the results are positive. So it's one of those things that my jaw just dropped because we don't usually get that. That's the translational approach. So that's my moment. It's too good of a moment, and I summarized it very poorly here, but it's too good of a moment not to share, even though it distracts from ALS. It's one of those, this is why I get up in the morning. This is why I do it. And it might never happen again that linearly, that easily, but just the chance that it might. Isn't that fantastic? We went from nothing to there's a drug.
Yeah.
And it's just, it blows my mind every single time.
I'm
so
sure
that's,
it was beautiful.
That's the answer that we have so far. And of course, we're not involved in any of the drug administration. That's the doctors make that decision. We found that it worked in cells. That's all we found. But they measured the risk success profile, and they thought it was a good choice. And apparently, it's working out.
Your lab is part of the people who are laying the foundation for the treatments, right? And from one person, would like to look at this because a lot of people, it's very hard to explain what you are doing in lab. And that's the objective of the show, that why this matters. It really matters for these moments like you just described.
It matters. It's impactful for us. It's impactful for others. We do what we do because of children like James. We do what we do for the ALS patients. That's why it matters.
Yeah.
Otherwise, we could be doing anything else, right? And not all science needs to matter for the same reasons, but this is why it matters for us.
Yeah. Thank you for your great insight. I learned a lot today, and I hope my audience do too. And it was an honor having you today on our podcast. Thank you again for the
Please,
time.
it was a pleasure to be here. Thank you so much for having me.
Thank you so much.